Is everyone able to see my first slide?
It's perfect, yes.
Amazing. Great. All right, so here we go. So super resolution, as you'll all know, is the general umbrella term for a number of techniques by which you can achieve resolution below the diffraction limit, which is a couple of 100 nanometers. And there are, broadly speaking, 3 ways to do this. You can make the incident beam size smaller, the effective size of the incident beam smaller, which is the approach taken by STED microscopy. You can access higher frequency information in wide field images by projecting patterns into a wide field image. And this, of course, is a technique that Reiner is particularly famous for. Or you can separate fluorophore emission in time and localize individual fluorophores. Now, localization microscopy has become particularly popular because, in many ways, it's the technologically simplest technique. You just shine a really strong laser onto your sample, and then if you get the right combination of sample and fluorophore and everything, you can get the fluorophores to spontaneously blink and you can localize them. But it has the combination, which is different to STED or SIM, where it's very simple experimentally, but it's still quite complicated in terms of the data analysis. And this is what particularly interested me when I entered the field, because I'd very much specialized in data analysis previously, though I had done quite a lot of experiments. So I was very intrigued by this combination. Now, when I came to the Randall, which is where Reiner and I were at the time and where I still am, I was very interested by what this new technique could do for cell biology. And so I went round to various cell biological colleagues at the Randall to ask them, what should we be doing as microscopists to try to get a better technique that can answer your questions? And this was obviously my first experience in trying to talk to biologists about technical requirements, because I got a very long list, and of course it wasn't all achievable. The ideal technique, they said, should be high resolution in all 3 dimensions with rapid image acquisition, should be usable in live cells with existing fluorescent proteins, shouldn't require an expensive microscope, and shouldn't require complicated data analysis. And what I've realized over the intervening 10 years or so is that you can't always get what you want, but in the immortal words of the Rolling Stones, if you try sometimes you might just find you get what you need. And this is really what this talk is all going to be about. You know, although obviously I'm going to have a focus on artifacts, it's really about also judging how to understand your data and how to set up your experiments so that you get what you need out of it. And of course, this being a super resolution talk, we're going to start out by focusing on resolution. Now, when I started out in microscopy, I felt like I understood what resolution was. It's fairly easy to show people 2 blobs and then have them move together and then see at some point they disappear into each other and you can't see them anymore. So this is a pretty easily understandable way of describing resolution, and you can write it down in the immortal form of the Abbe limit, which we are all, I would think, very familiar. And for some other techniques, you can also describe how you would get below the Abbe limit in a fairly intuitive way. For example, for structured illumination, for linear structured illumination, we know that we get at best half this value. For stimulated emission depletion microscopy, we can get a more complicated version of the resolution, which is to do with the power of the depletion beam. But as somebody who works in localization microscopy, as time has gone on, I've started to feel more and more like Spock from Star Trek, encountering some weird, previously unencountered human emotion like love. What is this thing, I say, that you humans call resolution when we come to localization microscopy? Because for localization microscopy, resolution actually varies locally. It varies with the properties of the fluorophores, it varies with the distribution of fluorophores, and with the knowledge that's used in modeling the data. So it's not nearly so simple a concept, and it doesn't have a single blanket value that you can apply to an image in the same way. And I found this a fascinating concept all through my time in localization microscopy. Now, localization microscopy is one example of a more general class of techniques where we can exchange knowledge for resolution. So this goes back all the way to the 1980s, where people first started to use image processing for improving microscopy images. And this is an example from back in 1983 by Agard and Sadat, where they used images of a zebrafish embryo, and I show the unprocessed images on the bottom, and they used the fact that they knew how out of focus light propagated through the sample, and they were able to remove out of focus light from the images and get back images that were much sharper. And I'm showing those 3 processed images on the top row. So here they were able to remove that out of focus blur that so often degrades wide field images, where even though the resolution is good as such, the information quality is very poor. And just to explain what deconvolution is, since this is a tutorial, I thought I would go through it. In image processing, we often demonstrate these things with a very famous model, though her usage has decreased in the last 5 years. It was originally normally demonstrated with a Playboy model who the computer vision researchers said had great texture, but I personally feel that David Hasselhoff has much greater texture, and so that is whom I will be demonstrating deconvolution with. So we start out with David Hasselhoff and David Hasselhoff's Fourier transform. And for anyone who hasn't come across Fourier transforms before, a Fourier transform is a representation of the different spatial frequencies in an image. So higher spatial frequencies correspond to higher rates of things changing, to things being sharper, to higher resolutions. So we can see in this Fourier transform, so the bottom image on the left-hand side, that we have the most information, the highest intensity, the whitest pixel in the center of the image, and then it decreases as we go further out. So there's the most high frequency, the most information is high frequency, and then there's less and less information as we go out to high frequencies. Then what I'm going to do, since I'm imaging this in a pretend microscope, is I'm going to convolve with the point spread function. So the middle image on the top is a microscope point spread function, an approximation of it. And on the bottom, I use the fact that in Fourier space, a convolution in real space corresponds to a multiplication in Fourier space. So in Fourier space on the bottom row, I'm taking the Fourier transform of David Hasselhoff and multiplying it by the Fourier transform of the point spread function. And what I get out, which is the 2 images on the right-hand side, is a blurred image and a Fourier transform which has less information at high frequencies. And that's why the area outside is all black at high frequencies. And now if I want to reverse that process, so I've convolved and now I want to try deconvolving, and this is what you do when you do deconvolution, you're trying to get back information. If I did the most intuitive thing and said, well, in this image I was multiplying on the bottom column, so the obvious thing you can do to undo multiplying is dividing. If I go back along that bottom column and then I'm going to transform into real space, you can see that in that bottom left image, unfortunately, I don't have very much information. It's all noise, and that's because we have a challenge that there is very little information in that outer area. So for most of the high frequency, for most of the high spatial frequencies that we can see, which are the black areas in the bottom right 2 images, we're dividing one very small number by another very small number, and therefore we're getting very high noise. And this is this general problem that will be particularly familiar to anyone who's done SIM, which is how do you retrieve high frequency information? You have to, with SIM, you transform it into a passband that's actually more visible. And in this case, what can we do? Well, we can do a very simple thing called Wiener filtering. We just put an offset outside on our bottom middle image, and that means that when we divide by it, we can get back an image which is much sharper. So if we compare the bottom left to the top right image, you can see the bottom left image is much sharper. It's got a better signal to noise, and particularly features like the gaps in the board that he's holding are much more distinct. However, you'll also notice that there's a distinct stippling effect. I think the German term for it is hammer pattern, and that's obviously very descriptive. It looks like hammer pattern, and it's at the scale of the point spread function. And this is a very common issue with deconvolution. It's hard to avoid, but it's also something that's worth thinking about when we start to do localization microscopy, that is very easy to introduce unintended effects when you do any type of fitting. Okay, but obviously we didn't just stop at removing out of focus light. We actually tried to, by the 1990s, people were trying to model the point spread function in this way. And this is an example from 1995 by Carrington et al., where they used images of microtubules, and then they used their knowledge of the shape of the point spread function to try and undo the blurring that the imaging had done. And I think that this is another really useful example of what this does, what happens when you're trying to build in more information is that you also build in more assumptions. So here we have what look like continuous microtubules. We think we're going to model them as being formed from a point spread function, but in the paper there's actually a key note which tells us what assumption is building in, which is we model the imaging process as consisting of a finite amount of data formed from a continuous object. And it's that phrase continuous object that is the key to understanding what's going on here. What we are assuming there is that there are no breaks in the structure below the diffraction limit. There are no separations between microtubules which are smaller than 200 nanometers. And if there were, then we would simply model the whole thing as continuous. So you can actually see here that we're getting back things which are separated by only about 100 nanometers. But we've built in this assumption that means that if that isn't matched, we could get back something that doesn't resemble the real sample. Okay, I've said all of that. Let's go on to what localization microscopy is. So localization microscopy is a technique where we separate fluorophore emission in time and then localize. So it relies on the fact that a feature of a known shape can be localized to a much higher precision than the resolution of the system. So the resolution of the system is 200 nanometers, but the accuracy with which we can find the position of a single fluorophore is not given by 200 nanometers. It's given by the fact that we can fit the point spread function, which I'm here showing the data in blue, with a model of the point spread function in red. And while the resolution of the system is given by the width of that point spread function, the accuracy with which we know the position of that one fluorophore is given by the mismatch between the data in blue and the fit in red. And this is given by the Fisher information limit, which is explained by a paper by Raymond Ober very nicely. But there is also a rough approximation, which was published by Thompson et al. In 2002. And that tells you that the accuracy with which you know the position of a fluorophore is given by the number 1 over the square root of the number of photons. And then there's also a component from the background, which kicks in surprisingly early. So in general, the more photons, the more accurate, but it's only increasing by 1 over the square root. So you do need a big increase in the number of photons to get a big increase in accuracy. And of course, one fluorophore isn't a picture of a sample. So most biological samples aren't just one fluorophore big. They are made up of many, many fluorophores. And so we build up the image over thousands or tens of thousands of frames, in each of which only a few fluorophores are localized. So, of course, this is simulated data, but in real data, the process is entirely similar. We're building up the image. Now, if I just go back and do it again, you can see that at the beginning, even though each fluorophore is localized very precisely, you don't really have a good image of the sample until you've built up a large number of fluorophores. And that brings in another important factor that it took a long time to think about, and we'll come back to later, which is the number of fluorophores which you have imaged is just as important as the precision with which you can localize the individual fluorophores. But of course, that's also heavily dependent on what the sample looks like, and that's a really intriguing mathematical problem that we'll come back to. Of course, most fluorophores don't actively photoswitch like this. I'm not going to go into detail about this here because this is going to be much more focused on the analysis side. But just a note, you have to be able to achieve blinking. You have to have fluorophores switching on and off. And there are, broadly speaking, 3 ways to do this. You can have fluorophores which exhibit photoswitching behavior. So that's the basis behind Palm and Storm. So you have one channel which allows you to switch, one laser channel which allows you to switch, and one laser channel which allows you to excite the fluorophore. The second method, which is probably the most popular method today, is using non-photoswitchable fluorophores with a reducing agent and a pH. So you put a reducing agent into the buffer. You create a buffer with a combination of a reducing agent and a pH, which induces blinking. So Alexa 647 is the absolute classic dye. It's incredibly popular. None of the other dyes really quite perform to Alexa 647's level, but you can certainly do multiple colors. And thirdly, binding and unbinding of diffusing probes. So this is a method where you use DNA origami and fluorophores bind and unbind to the sample. And this has the advantage that you can use a single color channel and you're not limited by the photodynamics of the fluorophores because it's all about how it binds and unbinds. So I'm just going to check. Are there any questions so far? I'm going to just switch back. No, I'm going to go back to my...
The audience cannot actually have the audience on except if I allow them, if I have to move them. So they can ask questions in the chat, but I will let you know if there's a question.
Great. I will go back. Am I back on my presentation? Okay, I'll assume I'm back on my presentation. So thanks, Rainer. Feel free to ask any questions in the chat. Rainer will alert me. Okay, so let's go through some of the basic processes which we would need to go through if we were going to do fitting of localization microscopy. So I'm really going to break it down into some of the very basic tasks because I think it's really helpful to have an understanding of what types of things these algorithms are doing, because it then allows you to work out whether you are likely to be getting errors. And then a little later on, because we do have quite a long 2-hour slot, we'll get on to how you might identify those errors. Okay, so we have a few fluorophores on, and obviously in this image that I've given you, I have a few fluorophores which appear to be single, and then I have what looks like a pair. So I'm going to demonstrate an algorithm called flood fill. So here we firstly simply threshold out all the areas. And flood fill is an algorithm for identifying contiguous areas above a certain threshold. So it identifies areas where the pixels are joined to each other. And that's important because we're trying to identify all the pixels which might have come from a single fluorophore so that we can then fit them. So how flood fill works is you choose any pixel. I'm marking it in red. You select any red pixel and mark it as blue. Obviously, these are not real colors, they're just designations. You select all the white neighbors of that pixel and mark them to red. Select any red pixel and mark it as blue. Set all white neighbors to red. Select any red pixel and mark it as blue. And we go around and we just do that simple algorithm over and over, selecting red, marking as blue, over and over. And eventually we have no red pixels, and so there's nothing to change. And so at that point, we know we've identified a contiguous area of the image where all the pixels are connected to one another, but they're not connected to anything else. And this is what we would then fit as arising from a single fluorophore. And then we go and we do it with the next area. But I'm sure you can see that what this cannot do is make any intelligent decisions about whether this is a suitable area to fit. So that has to be done either by the thresholding or by something else in the algorithm. Here we are simply identifying areas of contiguous pixels. Now we're going to just talk about a couple of simple methods that you can use to fit your data. The very simplest is centroiding. And it seems so simple that it's impossible it could actually be effective, but it is actually pretty good. All that centroiding is a weighted average. You just take each of your pixel values, you multiply it by the intensity, and then you correct by adding up all your intensities, and that is it. That is how simple it is. And astonishingly, it actually does pretty well. The only thing that's a disadvantage of centroiding, it of course doesn't take shape into account, so it's not the most accurate. Also, it can be a little prone to an effect called fixed pattern noise, where you tend to, you have a bias towards certain regions of the pixel when you do your fit. But overall, it's very impressive for how simple it is. Probably the most common method is fitting a Gaussian. And here I'm showing an example where I've got the data in blue, your current best fit in red. Obviously, my starting current best fit is pretty bad. The contribution of each data point in green, and then the contribution to mu in white. And I go back and forth. And as you can see, I'm getting a better and better fit where my error is getting smaller over time. And you can see eventually I find a mean value, which is pretty accurate. It's where you would think that the center of the peak is. So fitting a Gaussian is very accurate. It's very fast as well because of certain properties of a Gaussian that let you do the fit very fast. I would say the only disadvantage of the fitting of a Gaussian is, well, I suppose there are actually two. Firstly, the point spread function is not a Gaussian, so it isn't actually the most accurate fit, and there are certainly some interesting approaches that have been used recently, using things like deep learning to learn the point spread function, or using cubic splines to do a more accurate fitting of something that is more like a point spread function. So there are more accurate approaches, but a Gaussian does still perform really well. The second disadvantage to a Gaussian is related to the properties of the Gaussian in terms of how different it is when you have one Gaussian as opposed to two Gaussians. And Gaussians have a property where if you have two Gaussians on top of each other, if you fitted it with one Gaussian or with two Gaussians, it's an incredibly small difference between each of them being the correct fit. It's less than 1% for cases which we've tested extensively, and this is much, much smaller than the contribution of the noise. So because of that, it can be really, really difficult to tell whether you have one or two Gaussians. And this is something that we are going to come back to a lot later, because obviously it's very important, if you want to fit the position accurately, to know how many fluorophores are contributing to a given patch. And then something that I'm not going to talk about a lot later, but if you do localization, you will do a lot of, is drift correction. Now, because the data sets are so incredibly long, you might be collecting for 5 minutes. People generally do drift correction. This is an example just with some beads as a correction. And you can see that we're able to fit the position of the bead and correct the position of each frame very nicely. The only disadvantage is you have to put beads in your sample, which is obviously not particularly desirable for a lot of people. So a lot of people now do cross-correlation based drift correction. Now, you do have to bear in mind when you do that your drift correction will be limited by both the photon count of your images and by how similar your images are when you project down the number that you're going to do a cross-correlation on. So cross-correlation just tries to find the best fit in terms of how offset two images are. So generally what people do is project down 1,000 images or maybe even a few more, and then in those chunks they ask how much has the sample moved in between each one. And this really depends on the sample appearance as to how accurately you can do it. The final thing that I think we should think of before we move on is something that I think is worth always thinking about when you have an image, which is your brain is very good at creating patterns. It will try and close structures which are not continuous. It will see structures that are not continuous as continuous, and it will assume that things continue behind each other if you see one being occluded by the other. All these things are things which may be true, but are not necessarily directly supported by the images. So it's important to remember when you look at your images and see something and you think, aha, this shows that this is a combination of what the image actually shows and your brain's interpretation. Now, there are quite a number of very nice free localization analysis softwares. This is a few years out of date. There's some really there's some good ones on the market now that are on the market. There's some good ones available for free. Thunderstorm is probably still the most popular one. It's incredibly stable. It's unfortunately not really being developed anymore, but it's still very stable and very functional and good for both using and if you want to develop your own things that use that. But if you want to know more about how well these perform relative to each other, there's been some excellent work by Daniel Sage and colleagues on how the different localization microscopy softwares perform relative to each other. And that's been published twice in Nature Methods. So we know that knowledge is proportional to resolution. And that's really what I've been trying to get across in this initial section of the talk. That in localization microscopy, the amount of knowledge that we're able to build in and the accuracy of that knowledge yields us resolution improvement. But it's really important for us to know what's the exchange rate, how much knowledge that we're able to give them, which gives us what improvement in resolution, and how do we measure that? We want to know what the resolution is when we get an image. But since the resolution will vary with each experiment, ideally we want a measure that should work on any data set. And in 2013, we seem to have got that. We got this method called Fourier ring correlation analysis. And this was developed by Robert, oh no, I'm going to say his name wrong. such an excellent thesis as well. Robert Nieuwenhuizen and Bernd Rieger, and at the University of Delft, and they did a really great job of translating this method that was so widely used in electron microscopy into a form that could be used for localization microscopy. Now, what Fourier ring correlation does, which makes it so special and useful, is instead of trying to measure how something performs relative to, say, a test structure, which is the easiest way to measure resolution, but biology doesn't entirely consist of test structures, so it doesn't obviously work for a random experiment. What Fourier ring correlation does is it asks how similar is the image to itself. So instead of asking about how similar the image is to a test structure, you're asking how similar is the image to itself. Now, obviously, to ask how similar the image is to itself, you need to have some way of creating two images. And the way that they do that is shown in this figure in the part labeled A. At the top, we see one image, and then we take each of these localizations that are present in that top image, and we randomly assign them to two images. So we see, we start out with an X shape, we get two X shapes where each localization from the top appears in one of those two images. We then take the Fourier transform of each of those images, and we see that on the third row down. And we then ask for those two Fourier transforms at each length scale, that is, for a ring centered in the middle at zero spatial frequencies, for a ring that starts at the center and then expands out, at what radius of the ring does that image start to be not correlated? And not correlated is something where essentially you have to introduce a number. It's an arbitrary number, but it's a number below which we say we can't see something. Now, that might seem a bit arbitrary, but if we think back to the Abbe limit, we're actually doing the same thing there. We're taking two peaks, we're bringing them towards each other, and we're saying at a certain point, the dip in the middle isn't big enough for us to tell the two apart. And that's an arbitrary distinction as well. So we have this relationship on the bottom. You can see that we've got here different spatial frequencies. So that's moving out from the center on the Fourier transform images. And we can see that the correlation between the two images is falling off from 1, which is perfect correlation, and getting lower and lower. And then at a certain point, we say, okay, at this spatial frequency, we don't think the images are correlated anymore. That corresponds to a certain distance. And we say that that distance is the resolution. Now, it's important to note that this resolution definition doesn't directly correspond to the way that resolution is measured in the Abbe limit. So it is actually going to give slightly different values to the values that you might see. And certainly, I think you can see in Bernd Rieger's original paper that for if you take into account all the things that can possibly bias a Fourier ring correlation measurement, particularly repeated localizations, which are quite hard to take into account, but do tend to boost the resolution a lot, because then you're measuring the precision of the single molecule measurement rather than the resolution of the whole sample. If we do that, then he was getting measures of about 80 nanometers for a high-performing dye like Alexa 647, whereas normally if you quantify that using resolution measures where you try and measure sharpness or separation in some way directly in the image, you would maybe expect to get about 30 nanometers. So it does tend to give you about a factor of 2 worse in terms of the values that you get. But on the other hand, it's really the only way that works with data and that isn't just drawing a line scan on the image, which was not really very satisfactory ever. as I say, the disadvantage you have to take into account is that it can be very biased if you have single fluorophores appearing multiple times. Now, Ben did create a method to take this into account in the original paper, and it does work, but it's often not implemented in the versions of Fourier ring correlation that are available via plugins. So if you see values in papers that are much smaller, you do have to bear in mind that it might be taking those repeated localizations into account, which will then improve it by a very big factor. So to give an example, we've found in our experiments, when we didn't take the repeated localizations into account, we would get values of about maybe 15 nanometers. And then when we put in the maths to remove the effect of the repeated localizations, it went to about 75 nanometers. So it is a big factor difference. Now on the right-hand side, Bent is going through a few interesting effects which you can quantify when you use Fourier ring correlation, which was really great. He was able for the first time really to look at the effect of the labeling density on your sample in a really quantitative way, and the localization uncertainty. So both the localization uncertainty and the labeling density have a really strong effect. But this also intrigued me when I saw it, because of course the labeling density kind of has different meanings in different types of sample, because here he's saying 10 to the 4 per micrometer squared, but if you have a very thin linear sample, you might have locally fluorophores which are only a few nanometers apart, and yet you might not achieve this labeling density. So this was something that started to percolate away in the back of my mind. Now, of course, I said that I was going to talk about artifacts, and there's a lot of that coming up, but I first want to introduce that idea here. There are a lot of possible ways that you can get artifacts in localization microscopy images, and I just want to touch on some of the ways which are not analysis-based first, because obviously in my career I've been quite focused recently on the analysis-based artifacts. But there is also fixation as a step, which can really change both the global and the nanoscale structure. And there was some really great work by Sylvia Ritzley done in 2017, looking at the impact of different fixation methods. It's hard to quantify across all different cell structures, and the right fixation seems to vary a lot on the type of cell structure that you want to see. But it's very important, and it can completely change the results that we see. Also, of course, the analysis can lead to a lot of artifacts, and that's something that I've really been obviously bringing up throughout this talk. But here's a first example where I've just done a very simple simulation. We've done just a line and then a cross line at low density data where the fluorophores are all well separated, medium density data where we have a bit of overlap, and high density data where most of the fluorophores overlap. And in this case, of course, we know what the ground truth structure is, so we can see what's happening. And you can see that in the low density data, which is in fact the best fit, the data are somewhat scattered. Then in the medium density, the line appears a little sharper. The crossed lines, we start to get a bit of disappearance of the middle bit of the cross. And then in high density, the line is much sharper at the ends, but the middle has disappeared, and the cross, the middle has completely disappeared, and we just have very sharp bits at the edges. And this starts to hint at one of the challenges that we're going to come back to later on, which is that when you have an artifact, when you have an inaccuracy, you're not necessarily getting further away from the ground truth position. So if we look at the high density images, either of the line or the cross, the parts of the image that are present are actually closer to the ground truth structure in this case, because they are, we have a line, and when you have multiple things on a line, you tend to bias your fits towards the mutual center of overlapping things. And if you have a line, that mutual center is along the line. So the structure which is present is closer to the ground truth structure, but you have a lot of missing structure and your fits aren't actually accurate. So if we had, for example, a double line structure, we would lose that completely, while in the low density, we would still get something that reflected the size of the sample, even if we couldn't see the structure. So I want to bring up something that we're going to bring up again later and think about in more detail. A couple of years ago, there was the first real attempt to do error mapping in localization microscopy images. And this was called Squirrel and was developed by Sean Cully at UCL. And it's very much something that inspired us. It was a really interesting approach. So let's just think about what they did, and then I'll discuss later the sort of advantages and the limitations of this approach, so what it can give you when you try and assess your own images. So what Squirrel does is it asks, how similar is the localization image that I got to the wide field image? So if we look on the top, you can see that what the method does is it takes in a super resolution image and a reference image, it aligns the two, and then it creates a new reference image with the super resolution image, and it asks how similar is what I get out if I convolve the super resolution image with not exactly a point spread function, but something similar, and then I compare to the reference image. Now, this can give you an error map. It's really great for showing you missing structure, and it shows you how linear the relationship between the wide field and the super resolution image is. So this is a way that you can get maps of how much your super resolution image is differing from your reference image. So we're just going to put a pin in that, and we're going to go back to this idea that I brought up at the beginning, that people wanted methods that could be used in live cells. But this, the limitation that we're going to be tackling could equally be being able to use multiple colors easily. For all of these applications, what we have the challenge of is, how do you deal with data in which multiple fluorophores are overlapping? And this is a challenge even in very low density data, because fluorophore excitation is a stochastic process. Even in very low density data, you are occasionally going to get two fluorophores that overlap. And it's a particular problem when we tend to look at biological structures where the fluorophores distribution is very clustered. We're not often looking at something where the fluorophores are just uniformly distributed across the whole image. We're usually looking at things where we have quite strong clustering or grouping of fluorophores, and that means that the probability of the two fluorophores will turn on and overlap is quite high. So if we do want to make it faster, in this case, we end up with exactly this problem. In ideal data, you have low density and well-separated fluorophores, but this limits their speed of acquisition. So what happens if I increase the density, spots overlap in the image, the localization algorithm thresholds these out, and eventually no fluorophores can be localized. So if this is what our original localization microscopy looked like, what happens if we think about changing the activation density, that is, the number of fluorophores that are activated in each frame? So we start out here with very few fluorophores being excited. And you can see as I increase the activation density for a while, I get more fluorophores being localized on the right. I'm filtering them out after a while so that we can just see how many are being localized for a given activation density. But it increases for a while, but then as I increase the activation density further, it decreases again because fluorophores are overlapping and I'm not able to image them successfully. So this was a big issue. It also comes up all the time for two color measurements when you're doing D-storm with multiple colors. What do you do when you're trying to localize two colors? When you're trying to localize something where you have overlapping fluorophores. Then I started to think about the fact that I brought up when we were discussing Fourier ring correlation, that it's how likely you are to get overlapping fluorophores also depends really strongly on the sample. So being a physicist, I started out from a very simplistic view of thinking about three different types of sample: a zero-dimensional sample where we had just very small clusters that were well spaced, a one-dimensional sample where we have something linear like microtubules, and a two-dimensional sample where we have something that's distributed at the scale of the point spread function, is distributed over 2 dimensions at the scale of the point spread function. And that could be something like a focal adhesion. And I activate all those types of samples at a given number of fluorophores per unit area. And then I ask, how many successful fits can I get per sample area? So on the left, we can see that we have, I'm showing on the bottom row in yellow the successful fits, and in blue the fits that wouldn't be likely to be successful because the fluorophores are too close together. And you can see that the number of successful fits actually goes down as we go to a 2D sample because we have so many unsuccessful fits in the 2D sample. So we're activating with what would be the same laser intensity, but what we're getting out is we're getting, for the 0D, a lot of successful fits and a few unsuccessful fits. And for the 2D sample, we're getting almost all unsuccessful fits, and we would just have a very small number of fits that would be okay. So this suggests that there's going to be an incredibly strong effect with the sample structure on how fast can you image. So I became really interested in what the theoretical limits were for how fast you could go and how you could transmit information through the microscope. Because it's almost like you're trying to transmit information while wearing a huge rubber tube around yourself. You're trying to get the information through, but you have to do it while obscuring things with the point spread function. And that's because the point spread function affects the number of fluorophores that you can't image, and the structure of the sample changes the number of fluorophores that you can't image. So here I'm showing the 2D sample on the left, 1D in the middle, and 0D on the right-hand side. And in red, I'm showing the fluorophores that cannot be imaged when we are imaging the fluorophore indicated by the green star. And you can see that the number of fluorophores that you cannot image changes very rapidly as you go from 0D to 2D. So we decided to see if we could reproduce this, and we found that you could change the imaging speed. You could quantify how that would change the imaging speed. So I'm showing the activation density on the bottom axis and the number of images that would be required to attain a given resolution on the y-axis. And you can see that when we have a 0D sample, which is in black, we actually don't require that many images at all, maybe, maybe 10 or maybe sort of 30. And then when we go to 1D, we need maybe more like 500. And then when we go to 2D, we need more like 10,000. So this is a very, very big difference. So you're looking at a factor of 10 to the 3 difference to go from 0D to 2D. Oh, no, sorry, blue is 0D and black is the 2D. I read the graph the wrong way around. Okay. Can we see the speed difference in live cell imaging? So the first thing we wanted to know was, can we see what difference, this difference in the acquisition of a certain resolution in live cell imaging? And here I'm showing on the left the Fourier ring correlation resolution and then I'm showing how many images, the number of images we take, and what Fourier ring correlation resolution we achieve with those images. And you can see that the image resolution is getting better as I acquire more images, but between 2D and 1D, we're actually getting about a 30 times speed difference to get a given resolution. And in this case, what we did was we went a little further back from the ideal activation density in each case. So we were at a similar activation density compared to the optimal one. And then we thought, well, we've shown this in a real sample. What we should try and do is to quantify these results, which I was showing, which we simulated using Fourier ring correlation. And this is where it all went quite wrong, because we assumed that we would get a nice graph showing a turn-up, and we actually don't get that at all. What we see is the resolution that's reported just gets better and better and better as the activation density goes up. And that's because Fourier ring correlation doesn't distinguish between sharpness and true resolution. All it can measure is the sharpness, and the image is always getting sharper as you go to higher activation density. As we showed before, as we increase the activation density, the image always gets sharper, and any structure that wouldn't get sharper just disappears. So we know that Fourier ring correlation won't give you a warning. So Fourier ring correlation, when you get sharpening artifacts like this, just gives you a higher resolution. Estimation methods don't give you a warning. They also just give you a higher resolution. So if you try and estimate from the number of photons reported, that also reports a higher resolution. We don't generally have ground truth available because normally there's no ground truth data. And it's very difficult to assess the density of activation, which is what we tried to do for a long time, because if the density of the data is too high, there will be many misfits, and we don't have a priori knowledge of the structure. So we end up trapped in this loop where we don't know the structure, we don't understand what's going, we don't know what we're expecting to see, and so when we see something else, we're not able to tell what's gone wrong. So we first attempted to tackle this by doing a machine learning approach where a user tackles the problem of identifying poor fits. And we did this because it turns out even undergraduate students are far better at identifying poor fits than any of the automated systems that were available at that time. And that worked pretty well. It was able to classify using a classification that a person had input for that particular type of image, whether a fluorophore would be successfully fitted or not. And so we were able to take data like this and then reclassify with the good classifications in green. And you can see that the poor classifications in blue effectively take away the artificial sharpening that we see in this structure, which we know, since it's tubular, we wouldn't expect to get broad areas that are blurred over. And if we went back to our original models, in our original models, we had quite a big misfit between the simulation and the mathematical model. And we had thought that was not significant. Now, this was a case where we had a referee who really pushed us, who thought that there shouldn't be a difference between the simulation and the model. And while initially I strongly disagreed with them, it turned out that they were completely correct. When we removed the misfits from our simulated data, we actually got almost perfect agreement with our models. So that was great. So we've looked at reclassifying fluorophores. This is a way to deal with things if you want to get rid of anything that has overlap. But what happens if you actually want to try and fit those regions that have overlap? Can we trade greater knowledge for higher speed? And there are ways we can do that, and they're all centered around building in more information. Now we have two possible sources of information, either using our knowledge of what overlapping fluorophores look like to separate fluorophores in each frame, or using our knowledge of temporal variations to improve the resolution. So I'm just gonna, I think we have been going for an hour now. I was wondering if people might want a short comfort break of 5 minutes. 2 hours is a long time to just sit here. Or would people prefer to carry on?
I don't know. Can we have some sort of poll in the chat, or how can we find out?
I don't know. Would anyone like to say in the chat if anyone would like a short break? Hello. Hi, Danny.
Well, it looks like
break. Good idea.
Okay.
Okay. I'm just gonna stop my preview. So yeah, thank you everyone for sticking through the first hour. That was, I know it's a long time. Yeah, so if we just take a couple of minutes break and come back, and then I will take you on the exciting journey of what we can do when we have more information.
Okay, so let's reconvene at 10:23, okay?
Yep, that sounds great. Thank you very much. Oh.
maybe in the meantime, I can explain how it works with asking questions for those who don't know. So you put a question in the chat, and I can then move you to the stage, and you can ask your question live.
Yeah, and do feel free to ask at any point. I'm very happy to be interrupted by any questions. Yeah. So how are things in Jena, Rainer?
Well, basically fine, but I don't need to talk about the pandemics, right?
It's definitely still here. Yeah. Yeah.
And politics has decided to stop doing anything about it now, pretty much.
Well, I guess, yeah. I mean, excitingly, I have actually had the vaccine. So,
okay.
Do something. Yeah. It's quite exciting because, the Newhan's house is on a hospital campus, so they firewall all the medics by vaccinating everyone.
And then you're suddenly a medic now.
Well, I count as I might get in contact with medics and infect them. So, yeah. But yeah, it was pretty great. Though it did make me feel pretty bad for about 2 days. Just incredibly grotty. But I was very glad to have it.
Was it the AstraZeneca one?
No, I had the Pfizer one. So.
Okay, so I think we should continue the talk. So, Susan, stage is yours.
Great. So we'll just take a second to share my... All right. So do let me know again if there are any issues. Otherwise, I will assume it's displaying correctly. All right. So onwards with some tea now. So what I was saying before we had a little break, how can we trade knowledge for higher speed? So we have two possible ways to build in more information. We can either use our knowledge of what overlapping fluorophores look like to separate fluorophores in space, or we can use the fact that we know that fluorophores are turning on and off in time to improve the resolution. So there have been a lot of approaches for each of these methods. So particularly for using knowledge of what overlapping fluorophores look like in time, Dow Storm was a very well-known early one. There's also Fang Huang's multi-emitter fitting, and of course Ricardo Enriquez's SURF. For temporal variations, there's less methods that have been made, but probably the most famous is SOFI, super resolution optical fluctuation imaging, and this looks at the moments of an image. So actually, it's super easy for you to do a sort of poor man's SOFI, if you want, and you can do this simply by importing a stack of images into ImageJ, and then when you do a projection of it, instead of doing an average, if you do the variance or the standard deviation, then you will get a second order SOFI image, and that's the second order moment. And if you do that, you'll see that the image does become sharper and the background tends to become less visible. The thing that is important to remember with SOFI is that it is nonlinear in its effect. And this is in fact going to be a recurring theme with more or less all of these methods. More or less everything in localization microscopy is nonlinear. Ideally, you would just want to pick up every single fluorophore one time, but that pretty much never happens. So it is a nonlinear technique. There's also, you can also build in both temporal and spatial information, and this is something that, in fact, Rainer and I published together back in 2012, using a Bayesian statistical model of blinking and bleaching to model the whole process of fluorophore photo switching, and then to use that to build up a model of where fluorophores are in the sample. So we've always known that there were various trade-offs involved in deciding which imaging technique to use and what algorithmic trade-off to use. So in algorithm trade-offs, there's the acquisition time in your experiment. So if you do a faster acquisition, you're probably going to have to do more analysis. There's the assumptions that you build in. So if you build in a lot of assumptions, then it's going to be easier to do the fit, but maybe it won't be as accurate. And of course, there's the processing time. With the experimental trade-offs, there's the fluorophore choice, and there's whether you can go into live cell and fixed cell. But for all of these methods, I increasingly became concerned because all of the high-density methods have a disadvantage, and in that I completely include 3B, my own methods, every single one that I've tested. They all have this disadvantage. With some it kicks in earlier, with some it kicks in much, much later. But at some point, if you have too high a density of the raw data, all these multi-emitter methods fail, and they fail silently. That is, you will get a super resolution image, but this image is something that the algorithm has made up. And I show here an example where I've got thunderstorm fitting of, where I have fitting of a high density data set where I have two lines that are separated by 200 nanometers. So you'll note 200 nanometers is actually basically at the resolution limit. So we should definitely be able to see this if we're getting any degree of super resolution. But here I've done fitting of this high density data set with thunderstorm single emitter, thunderstorm multi-emitter, SOFI, and SURF. But I want to emphasize I'm not picking these methods out in particular. I'm just used those because they were popular examples in their respective areas. It absolutely happens with the other methods that we've tested. It just might happen at a different density. So with thunderstorm single emitter, we get a very sharp peak here in the middle. Thunderstorm multi-emitter, it's slightly wider. SOFI and SURF, you can see that we get with SOFI a very uneven distribution across the distance between the two lines, and we also get a lot of areas where we don't have any intensity. And then with SURF, we get a roughly even fill between the two lines. So I think I went through this before, but I'm just going to quickly run through again. How do you tell if you're getting this type of artifact? Fourier ring correlation won't give you a warning, because if you get artificial sharpening, it'll report a higher resolution. If you have any method that's based on estimating your resolution from the single emitter fitting accuracy, it also won't give you a warning. It'll also report a higher resolution. We, generally speaking, don't have a ground truth available, and it's very hard to assess the density of the raw, the activation density of the raw data. So I came to the conclusion after a while that what we needed was a way to be sure that what we got out was a real structure, even if the experiment needs to go fast, even if the density varies across the sample, and in some areas it's fine and in some areas it isn't. And as the developer of one of the slowest multi-emitter fitting, of one of the slowest high-density methods in the entire field, I knew that it needed to be fast, simple to use, and not too complicated. And this was where I was, broadly speaking, achieving independence as a researcher. So rather than doing it myself, I talked to my brilliant postdoc, Richard Marsh, and said, this would be a terrific thing for us to do, wouldn't it? And then he went away and did it, which made me feel very Picard-like, carrying on the Star Trek theme. And what he realized was you can actually use a super simple approach, Haar wavelets. Now, this is just multi-scale temporal filtering. It's a pre-processing step you can do on your data that can then be followed by standard fitting methods. So it's really going back to this idea of you can use temporal or you can use spatial. And we're really going into that class of using just the temporal effect. But instead of trying to do it in one step, we're taking out our temporal filtering, and then we're doing everything else after it. So we've got our input data, and then we separate out fluorophores, which are meant for 1 frame, fluorophores, which are meant for 2 frames, fluorophores, which are meant for 4 frames. And to give an example of what this looks like in practice, I'm going to show you some input data where we start out with I'm just going to wait for the video to start. Here we start out with 1 fluorophore coming on. You can see it appears first in the 1 frame, then the 2 frame, then the 4 frames. The second fluorophore comes on. It then appears in 1 frame, 2 frame, 4 frames. So this is how our method breaks down data in which you can see we have fluorophores overlapping into data in which, generally speaking, the fluorophores are well separated and much easier to fit. And you'll also notice we're getting rid of a lot of the background offset, which also makes it somewhat easier. So to give you an idea of how this performs in practice, what we have here is high density lines, high density simulations, and I'm just going to go forward because apparently this video doesn't loop, at different spacings. And you can see it is very high density, but this is definitely the sort of data I've seen fitted with high density methods. So we then created these lines at many different spacings and analyzed them with and without Hawk with a lot of different analysis methods. And here I'm showing 2 examples. The first that I've shown you a bit of already at 200 nanometers separation, and the second at 140 nanometer separation, just to give you an idea of what it looks like at the different separations. And you can see that when we use Hawk with both the single emitter and the multi-emitter, here I'm using Thunderstorm, fits, and those are the last 2 pairs of lines to the right in each set, you can see we get back that separation. Separating the fluorophores or emissions into more frames allows us to do more accurate fits. And even though, because we effectively have very high background here, because we have fluorophores overlapping all the time, so your precision, especially in single emitter, might not be terribly high, you do see that separation. You do get something that's roughly the right size. And you're not getting artificial sharpening. So we were very excited by this because it seemed to promise that you could get back very reliable structure. Now, of course, being physicists, what we saw was single pairs of lines are incredibly good for testing. I wonder if there are any single pairs of lines that exist in nature in biology. So we went back to our biological colleagues and of course, we all have them within our muscles. They're sarcomeres, and they are beautifully regular, and they are pairs of lines. And this is an example of T12, a motif labeled with Alexa 647. I'm showing some low-density data on the left and high-density data on the right. The low density was just acquired after bleaching. And this is a myofibril. We know from our electron microscopy studies roughly what the separation of these should be. So here we can show that when you have your low density, you get exactly the separation that you get if you have high density data and you use your Hawk fitting. But if you use other methods, you're tending to see a pull towards the center, and with most you don't even see 2 lines, even though you can see that separation is all the way up at about 160 nanometers. So this seems really useful. It does seem like it's going to, when we have high density, give us a more reliable output structure. Does it work at even smaller resolutions? So we then looked at M8, which is more like 90 nanometer separation. And yeah, we see the same effect. When we use Hawk, we're able to get back that separation. And of course, these are biological samples, so it's not super beautiful. They are disks, so they are continuous structures, so you definitely have the possibility of them lying one way or the other and obscuring themselves, so it was a much more challenging experiment. But you can see we could still see the 90 nanometer separations when we did Hawk, which is exactly what we also see when we did our low density experiment, 90 nanometers. So this seemed really promising. We were able to get back biological structures, which we know from our characterization with EM, what the separation should be, and we can also see that separation in our low density experiments. We also did DNA origami, which was down at 50 nanometers. Obviously, this is a much simpler structure. It was just 2 fluorophores that were well separated, and we were able to get back the separation of those fluorophores by using Hawk. Now, I'd started out by saying that what you might want to do is live cells, and obviously testing in live cells is pretty difficult. But what Richard had the idea of doing was using MEOS 3.2, which can emit in either the green or the red, and you can switch it between those states using the 405 nanometers laser. And what we were able to do was collect both low density and high density data from the same live sample, which was a really neat way of benchmarking what the performance of the algorithm was in real live cells as you might encounter them in a real experiment. So this is a partial tailing construct. We know what the structure should be. It should be a ring structure. It's in podosomes. And we can see that we're getting low density data pretty nicely on the left, and then very high density on the right. You can see that we're not really seeing any individual fluorophores on the right-hand side. We also did vinculin and focal adhesions, but focal adhesions, of course, being 2T, are much, much more challenging. And for various reasons, we only had MEOS 2 for this one at that point in time, and it was not as high performance. So when we analyzed these results, we were able to show the low density results on the far left, you see, are quite almost one might call diffuse. There are large areas that are white. We are getting not very sharp lines. We are getting a lot of structure where there is sort of a structure over quite a wide variety of distances. And we have structure like the bit coming off the little group of 3 podosomes that I've shown with the blue arrow. And if we look at the hawked image on the far right, you can see we see a very similar structure to that far left image. Now, of course, they were acquired over somewhat different timescales. It's not absolutely identical, but it is very similar. With the focal adhesions, we were able to acquire a lot more fluorophores for the focal adhesions, so we do have an image that's filled in over a wider area, but the structures are almost identical visually. But when we look at the alternative method, you can see that we're seeing a lot of sharpening when we use the multi-emitting fitting methods, and that's exactly what we'd expect from the fact that when you have multi-emitter fitting methods, you tend to have a silent failure at some point, and at some point the fitting won't be accurate. So we're getting sharpening, we're getting structures which are much sharper than they would be in real life if we were actually imaging them accurately. We're not seeing the structure as it really is in the sample. And for the middle, for Sophie, you can see here the structure looks more similar, but we are missing some of those fine structures, and we are also getting some background structure which isn't really present in the actual sample. So we thought this was really interesting and revealed some issues. We've done a lot more detailed studies, which I'm not going to include here because not everyone has my fascination with simulating circles. But you can actually have a look into, you might think, well, I might not care whether my image is sharpened. I might just care where the structure is. And there's a lot of interesting sort of detailed work you can do on how much each of these different methods actually biases the positions of different types of structure. And that's actually an interesting question and varies very strongly depending on the method. So all the methods show these effects, but as to whether they show strong biases in position, that's dependent both on the individual structure and the method. So this all seems good. So we thought, hurrah, we have a method that doesn't appear to show bias. So we published it. This appeared in Nature Methods in 2018. If you want to use it, it is available as a plugin. It was available with the Nature Methods paper. You can just run it. It's just a plugin. You put it in your plugins folder. You run your, you load in your data set, and then you run Hawk. And that is literally it. It takes about 2 minutes. And it is so simple that I got our Dean of Faculty at a live talk to demonstrate it, to show that literally anybody can do it. Because I think if our Dean of Faculty who can use it, anybody can use it. The only thing that you do have to be a little bit careful of is that a lot of the fitting methods, such as thunderstorm, have a tendency to fit single pixel events as arising from fluorophores. So they do tend to fit single pixels, not as noise, but as fluorophore events. And that can lead to this effect that I've shown here, where you have bright spots appearing at your original camera pixel distance. And this is just because for some reason thunderstorm cannot distinguish between single pixel events and fluorophores. So what you do to avoid that is just to do a filter which gets rid of everything above or below a certain point spread function size. So you only allow point spread functions that are roughly in a physically realistic range. So we usually use, say, 90 to 150 nanometers, and that gets rid of that effect very nicely. I would normally always do that for a reconstruction anyway. So I've made the case for why we might want this in fixed cells. Do we need in live cells? Do we need it in fixed cells? Well, I think it can often improve performance in fixed cells. The second color channel often doesn't perform as well as the first color channel. And this can lead to a lot of loss of information if you use a standard fitting method. If you use Hawk, you can get much nicer performance, much less punctate appearance of your sample. Now, I don't want to give the impression that we're saying that Hawk never goes wrong. We all know that any analysis method will go wrong at some point. And so this is, since I have this 2-hour slot, I thought I'd also show you how these methods fail. And this is the excess density failure mode of Hawk. And here on the top, on the left, I'm showing what the results look like without Hawk, and on the right with Hawk. And on the bottom, I'm showing what happens when you have very, very high density of podosome data of the type that we'd already seen. And you can see that Hawk doesn't necessarily get you something that looks super resolution, but it also doesn't look artificially sharpened. So Hawk is not going to give you an amazing resolution if your data isn't capable of that. But as you can see on the right bottom, what we see with the podosomes is we see what looks like a not particularly great wide field image. So if there is no extra resolution to be got out, Hawk should return you something that looks like a wide field image. And we actually tested this with Fourier ring correlation to see if Fourier ring correlation will then reflect the actual resolution. And it was really encouraging to find that it did. Fourier ring correlation told us that the resolution was garbage, and it looks like it's garbage visually. So that's a nice confirmation. And we can see from the quality of the data that we would expect it to be garbage. So garbage in, garbage out is often used as an insult, but in fact, when you have data analysis in science, you very much want it to be garbage out if it is garbage in. You don't want it to be garbage in, apparently nature-worthy publication quality image out. And this is just a similar image for the focal adhesions. You can see when we get to these very high densities, it just doesn't look on the bottom right, the Hawk image just doesn't look very good quality. Now, obviously, the next thing we'd like to know is, can you do this sort of correction in 3D? And we have tried a few things in 3D. We've tried looking to see if applying Hawk improves it. Now, astigmatism, this is very much sort of early results. Astigmatism is actually very hard because, as you'll remember, I said that we did size filtering, which is normally what you do for localization microscopy, but you can't do size filtering with astigmatism. And astigmatism is particularly prone to having things end up at one of the extremes of distance if it's incorrectly fitted. So while Hawk does improve it, let's go for some real data. While Hawk does improve it, here on the left I'm showing some 3D data without Hawk and then with Hawk, and you can see it does get better. It is still often not great. You're still ending up with quite a lot of fluorophores getting kicked to the extreme ends, and it is very hard to verify that it's really doing exactly what you want. Much better is the biplane approach, where we seem to actually get a really big improvement over the whole range and without any of the problematic aspects that you get with astigmatism. So this isn't published yet. We're still trying to work out the challenges with the astigmatism data, but the biplane data looked really promising. It was super simple, and it just seems to work. And here's some mitochondria. And again, we seem to just get an improvement with Hawk. And we can also test with that biplane data, varying the density. We had different densities at different points, and you can see as we go from low density without and with Hawk look pretty similar, up to high density where the Hawked image on the bottom right looks pretty similar to the low density image on the bottom left, but the no Hawk image on the top right looks very different from the no Hawk image on the top left. So this general approach can be used for some other things, which I'm just going to briefly talk about in case anyone is interested in how you can apply this sort of approach to other types of structure, where we can use the fact that we can convert some fluorophores and then monitor the other channel to track both the structure of the sample and single molecules moving within the sample. So this lets you track the global structure of the sample and also single molecule position. Okay, now I said that I was going to come back to Squirrel and how you can assess images. So as I said, Squirrel asks how similar is the method to the wide field image, and it gives a measure of how linear the relationship between the wide field and the super resolution image is. So it's a really useful thing to measure because it will tell you if you have missing structure, and it will tell you how nonlinear the relationship to the wide field is, which gives you an idea of how nonlinear your imaging is. But it's very important to realize that it won't be able to spot errors below the diffraction limit, so it's unlikely to spot artificial sharpening errors. So if you might have those, then Squirrel probably can't help you. And there's actually a really fascinating mathematical effect because we initially started to try and use Squirrel to look for artificial sharpening. But what we found was it would often classify the artificially sharpened images as better than the not sharpened images. And this is actually kind of what sent us down this whole rabbit hole of trying to whether we could do anything that would spot the artificial sharpening in the first place, because artificial sharpening is the main artifact that we deal with. So what we had to look at is we took some ground truth simulations, and then we tried blurring them. And what we then did is we looked at the structure present in those images at different length scales. So on the far right, I've got the ground truth. So I've taken the data in the ground truth image that I showed you here, and I've looked at it at different length scales, 25 nanometers, 50 nanometers, and 100 nanometers. And then we took, because we used very high density data we used a very high density data set, which we knew would give us artificial sharpening with one method, which I've just used surf here, but as I say, we could have used thunderstorm single emitter or anything else. And then we used Hawk single emitter, which we knew wouldn't give us artificial sharpening. And we then looked at the structure at 25 nanometers, 50 nanometers, and 100 nanometers. And broadly speaking, the Hawk single emitter appears to have more similar types of structure at those short length scales, which is what you'd expect because it should be most accurate. But we then reproduced what Squirrel does. We took the ground truth, instead of the ground truth, we looked at the expanded wide field of the surf of the artificially sharpened image. And what we found, what we found was that the ground truth is less similar to the surf than the expanded wide field. So if instead of the ground truth, I use the expanded wide field, which is what we use in Squirrel, we, at the 25 nanometer length scale, get something which is more similar to an artificially sharpened image. And that's because the artificial sharpening boosts these frequencies that are most dominant in the wide field. So it's a great method for looking at the linearity at and above the wide field, but if you need to go below the resolution limit, you need to take another approach. And that's what we think we've got a good way of doing now. And we've called this Hawkman. So it's really very simple. You just use Hawk as your reference image, which may seem too simple to even describe, but I'm going to describe it anyway because it was actually more complicated to develop than we thought, which is why it took us quite a few years. So you take your test image and your Hawk reference image, you flatten and blur them. We then took two approaches. We binarized and took a local threshold, and then binarized and took a higher threshold and skeletonized. Now you might think, especially if you do any image processing, why are you skeletonizing? Why? Well, obviously, nobody would ever skeletonize unless they had to, especially if you wanted to do anything quantitative. But the reason we did this was when we were looking, so unless you want to test for non-linearity, unless you want to test for linearity, which is what Squirrel does, and it does very well, but from using Squirrel, we knew that the disadvantage of doing that is that it will also pick up non-linearities which have correct structure, but are not actually not directly linear. So we wanted to take the opposite approach so that we would give as complementary information as possible. What we wanted to ask was not how linear is the relationship, but we wanted to ask how similar is the structure. And for this, we had to have some way of classifying structure and non-structure samples. So having these different thresholds and approaches allows us to classify structure in a way that you can't do if you're just looking at linearity. So if we look at the overlay, we then get two different types of map, one of which highlights to us here in magenta if we are getting any sharpening, and the second highlights differences in the structure. And if we blur and correlate, we get something that we've termed a confidence map. So we've combined the information in both the sharpening and structure maps to give a confidence map where mismatches are indicated in red here. So if we have a look at what sort of thing this can do, if we start out looking at different resolutions and what this type of approach can tell us at these different length scales, you can see that if we use a color scheme where red indicates full structure and blue indicates missing structure, you can see that as we get to finer and finer scales, we see the structure of the sample emerging. In effect, in the error maps, we can see the missing parts of the sample being highlighted in blue. We can see the full structure in the center of those two lines being highlighted in red. And if we zoom in, you can really see that the high density is localized there in the middle. the incorrect localizations in the center are being identified in red, and the correct ones are being identified, the missing structure is being identified in blue. Now, of course, we have to do each of these to a particular length scale. So what you get out is a number of different length scales where you're identifying missing structure, false structure, and true structure at each of the different length scales. And the length scale at which you get your mostly good agreement, mostly true structure, is your length scale of your resolution. Now, of course, this might not be a single number. It might vary across the whole image. So we had a look at some of the challenge data, and I'm showing here the single emitter and multi-emitter, both with and without Hawk. And you can see that for the single emitter and multi-emitter, we're able to see in the sharpening map at, I'm showing the sharpening map at 60 nanometers, and I'm showing you the structure map at 160 nanometers. In the paper, we show all the length scales if you want to see them, or it's very easy to reproduce because the plugin is available and the data is available. We just chose these as being the most illustrative and useful things to look at. And then we showed you the correlation maps at 100 nanometers. And you can see that, as you might expect, when you use single emitter, you get a lot more incorrect structure, a lot more artificial sharpening than you do in the multi-emitter. But even with the multi-emitter, we have some areas that aren't really accurate. And if you spend a lot of time staring at localization microscopy images, you could probably pick them out by hand, which areas you don't quite trust. And hopefully you'll agree that in the multi-emitter error map, we're picking those up pretty well. And this is just to show what it looks like when we go across those different length scales, starting from a very short length scale of 20 nanometers and going up to 200 nanometers, or up to 300. And you can see it hasn't even disappeared up at 300 nanometers. That's how much some of these artifacts stick around. So our method, this method Hawkman, isn't so much a resolution measure as it is a measure at which the artifacts aren't significant. But of course, we have also done it as a resolution measure, which I'll show you in just about one slide's time. Now, we wanted to quantify this on real experimental data. So we, again, because being a physicist, I'm extremely obsessed with how important the type of structure is and how that affects the information transmission. We did this on 3 different types of structure. We've got clathrin-coated pits at the top, microtubules in the middle, and mitochondria at the bottom. And you can see that we're able to identify those regions where there's missing structure or false structure. So here I'm showing the confidence map and the sharpening map. And you can see in both cases it's identifying problem areas that we can see visually very well. But you might be thinking, well, I could just look at the Hawk image. Why do I need, if you're just telling me I should always use Hawk, why should I bother using Hawkman? I could just use Hawk and not bother. Well, firstly, I accept that maybe not everyone would want to use Hawk. Not everyone always wants to use exactly the same method, but you might still want to know that your data doesn't have artificial sharpening. And of course, the second thing is there is a disadvantage to using Hawk. There's always a cost for everything. The disadvantage of Hawk is it does slightly degrade your resolution. So if you don't have any cases or no significant cases where you have overlapping fluorophores, then if you use Hawk, you will get a degradation in resolution for no improvement. So using Hawkman is a nice way of testing whether you actually need to do anything else. But we wondered whether there was actually something else, because there are still differences between the hawked and the non-hawked images, even when there's no visible structure. So what we did was we used a structure. In this case, this is a simulation, where we have 2 lines which are a certain distance apart, but the data is not good enough to show it in either the single emitter or the hawked single emitter image. So we can't see any structure in the hawked image, and we can't see any structure on the single emitter image. Can we get that does start to show you that there may be some missing structure there below the resolution limit of either super resolution image, that we're able to see this highlighted gap between the two outer areas, that we have some degree more confidence in the outer areas, but in the inner areas we're pretty sure that there is some structure there that it shouldn't be there. And we were then, of course, intrigued, could we see this in the real structure? So we used the Z1, Z2 structure, which is a very small separation of around 60 to 70 nanometers. Now, of course, normally one would be able to resolve that with super resolution, but we are operating at very high density, and the sarcomere structure, as I said, has a certain thickness, which tends to make all the separations less distinct. And what we found was that while we couldn't really see anything in either the Hawk single emitter or the single emitter images, we can see a small degree of separation at the very edges, but not in the center. If we look at the confidence maps for these small length scales, we are identifying that there should be two structures that are separated rather than a single uniform structure. So we are getting hints that we have an incorrect structure identified, that we are missing something below the resolution limit. So I think this could also be helpful. It could give you a hint if there's something that you're missing below your super resolution, and it could be a hint that maybe you could gain from optimizing your experiment. And of course, we do actually have in those maps information about the length scale at which the data is reliable. So what we've done is we've created an output map which identifies the length scale, the shortest length scale at which the data is in that small sub area is considered reliable. And so you can map your image as to the resolution at which it can be considered artifact free. And I think this will be quite helpful because it will enable people to test, are their images reliably artifact free at the length scale at which they want to draw conclusions. And this is freely available. It's on the bioRxiv where the plugin is as well. And you simply download the plugin and put it into your plugins directory in your image tray, and then you can use it. And it's, I think, pretty easy to use, but we are super receptive to feedback, and we would really love feedback. So if anyone wants to use it, if you have any issues, then do get in touch with us, and we would be very thrilled to hear back from you, hear feedback, and support you. We hope it'll be a really useful tool for the community. Now, obviously, I've been talking for nearly 2 hours at this stage, and I'm sure everyone has pretty much got to the stage of having enough, so I'm very much looking forward to questions. But I know that I've gone through an awful lot in this time about how we assess data using these different methods, and you might be feeling that this is all really confusing, and I've presented these different tools. When should I use them? So broadly speaking, I would say these are the situations. If you think you might have missing structure at scales around or above the point spread function size, and if you want to know if your super resolved image is linearly related to your output, then Squirrel is really great. If you think you might have artifacts in your image, or if you want to know if your data is biased towards by artificial sharpening, then Hawkman is a really great tool. If you just want to have a number for your resolution in nanometers because your paper is not going to get sent off until you have a resolution measure, then definitely use FRC. Just if you want them in number to be accurate, do make sure that you're taking into account multiple fluorophore reappearances. If you want to know more about Hawk, then as I say, do feel free to get in touch with us. The plugin is freely available, but we are very happy to hear from users and very happy to support everyone, especially if, as shown in this image, you feel that Hawk has knocked itself out. This is a hawk on an oil rig that was chasing a small bird and then flew straight into the oil rig and knocked itself unconscious. And to add insult to injury, the bird then landed on it for a picture. So if Hawk does this to you, or Hawkman, then do get in touch with us. We haven't had many issues, but we're very eager to solve those that there might be. And in drawing to a close, I would just like to thank the people who did all the work that I've shown here, particularly Richard Marsh, who led the work on Hawkman and who did so much great work on developing both Hawk and Hawkman from the very start, and Ishan Costello, who has done a lot of work on the 3D experimental side, and on getting data images for Hawkman, and Lisa Hirvonen, who really spearheaded the work on the podosome construct. Of course, I'd also like to thank all my funders and you for listening. Thank you very much.
Thank you very much, Susan, for this very inspiring talk. So I hope that there's lots of questions. So, there's already the first question. I don't know if you can see it yourself.
Yes.
So thanks for the tutorial. Although it's not a question. Thank you. So until the first question appears, can I ask you, if you look back at your first 3D publication, right? In what sense, would you say that some of the statements made in there have to be revised looking back now?
Yeah, so this is an interesting one, and I think it comes down to, I don't think we as a field, and certainly me in particular, really had a deep enough understanding of what resolution was at that stage. So I did actually go back and look in a lot of detail because I was concerned as to whether particularly the figures we presented for, say, the sizes of podosomes were accurate. And what we found was that 3B is really accurate for finding size and the position of the mean of those those types of structures. So the sizes and the average positions were correct, but I do think that there was probably some artificial sharpening, which wasn't really something that was discussed so much in the field at the time. So, I mean, I think that we were accurate in the sizes that we gave and in the size and the characterization of the structure, but yeah, I wouldn't do what I did then now. I would.
So the thread thickness is really not quite accurate, probably.
Yes. Yeah.
Okay, we have a question now. So have you tried to use image decorrelation analysis? And there's a link to a Nature article for estimating the image resolution. Oh. So somehow the connection let me see, Vitaugas, maybe I can put you to the stage. Where is it? So I hope that Susan comes back. You can actually speak now and discuss with her. Yeah, Susan is on her way back. Vitautas Navikas.
Yes, I'm sorry.
Hello. Here you are.
Sorry, okay. I tried to open the link and then close that form.
I was just really curious because this tool was developed by my colleague, basically the guys sitting in the same office, and we were like actually aiming to create something where you would be able to estimate resolution like for all the modalities from the one image.
Is this the tool from the Radonovich lab?
Yeah.
Yeah, sorry, I was trying to open it because I just wasn't sure from the description. Yeah, so we found, yeah, I should have actually mentioned that as well. Sorry, we even mentioned it in our paper. But when I was putting together the talk, yeah, I didn't get it in there. But thank you for bringing that up because I should definitely get that in next time. So yeah, the decorrelation analysis is, at least to me, and feel free to correct me, a sort of a generalization of a Fourier ring style approach, but with the generalization that you use phase information, and therefore it should be able to get back, what would you term it, a more accurate and general value of the resolution and whether there's any artifacts. So when we tested that approach, so we found when we tested it on simulated data, it was extremely accurate. But we found in experimental data that the information was often degraded enough that it wasn't really possible to get very accurate values for cases where you had sub-diffraction errors. I believe that the information in theory should be there, and when we carry out simulations and then use it on very clean simulation data, we are able to see it quite distinctly. But when we try it on experimental data, we tend to find that for the sub-diffraction errors, it's very hard in practice to pick them out using the decorrelation analysis. But I'd be really interested in talking to you about it because, yeah, it is one of those interesting things where we found it performed for us very, very well on experimental data, but as we went into on simulated data, but then as we went into experimental data, we found that the noise tended to overwhelm the phase information.
Yeah, that's definitely true, and we actually work on improving this. But I'm actually not the guy who is developing the tool.
But it's a really nice, and that was definitely also one of our inspirations, along with Squirrel. That I think a lot of us are thinking similar things. Wouldn't it be great if we could test, if we had some sort of general test that wasn't just FRC, which does tend to be so biased by artificial sharpening. And definitely in principle, that method solves this. But as I say, we find in practice, it does struggle with some experimental data.
Yeah. So, yeah, thanks for the answer.
Thanks.
Okay, thank you very much. Are there more questions? There's one more. I have a question about...
So we have also used it on podosome type structures. Yeah, so if you have, so this is a question about whether Hawkman would have issues with the skeletonization on nonlinear structures, and that's a really great question. So we have tested it on point-like structures, linear structures, things like podosomes where it's more blurred out and it's not really aligned, but it's not really a very extended area. And we have tested it also on mitochondria, which obviously also have quite extended areas of intensity. Oh, hello, Mohammed Reza. And we have found it performs pretty well. We haven't, obviously the skeletonization is something that we were a bit concerned about because it definitely has the possibility of producing artificial structure. And it was something that we were very concerned about, which is why we try and use it at a very specific length scale. We found it's pretty good for all those types of structure I've named. It doesn't seem to be a very big problem for focal adhesions. We haven't found it. It doesn't identify full structure in them. But again, you do have to be very careful about the length scale you use it at. And that is something that definitely, I know some people did not like the idea of using skeletonization. And for those people who don't want to use that particular type of thing, we also just provide the sharpening map, which doesn't use any type of skeletonization. So I know Bernd wasn't so keen on the idea of skeletonization. I can completely understand because it does have that possibility of creating new structures, apparently. We tend to find that we haven't found that happens in practice, but we would always provide the sharpening map to people. So if they don't want to use the skeletonization or confidence maps, that's still available to them. Sorry, your mic is off. We can't hear you. No, I still can't hear you. Sorry.
Okay.
Maybe. Katrin, do you have a question? You might need to type it. I don't know if we'll be able to hear you. We seem to have a few mic issues.
Can you hear me now?
Now we can hear you.
Hey, Katrin.
Hey, very nice talk, Susan. Very inspiring. I have a question because I lately turned more into translational imaging, and I was wondering, because that's exactly the point. In translational imaging, I feel it's more important the accuracy of the image than the precision at the end, because if you want to have diagnostic tools and stuff, you really rely on the fact whether that is true, what you see. So I was wondering whether you could maybe or is it possible to combine your very accurate like Hawk technology with artificial learning? So for example, to indicate in the image where it's maybe a good guess also by the artificial learning process, and where it's really analytically safe to see it. Because sometimes, you know, we have partly garbage in, but then you can heal it by adding another training mode, but indicate it in a color code, for example, that this is not as accurate as it is. And then maybe in the context of a huge tissue sample, you could still learn from it. I just have a problem with, of course, we as a scientist, as a physicist, we would like to have it as accurate as possible, but we have it also, I don't know, we can't leave black gaps, right? So in a tool.
Yeah, the question of how one might use artificial intelligence and deep learning to enhance data, and then how sure one can be that the results are accurate, is a very interesting one, and I think something that we are going to have to think deeply about in the next few years. I actually have a review of deep learning and localization under review at Molecular Biology of the Cell, and we'll see if that gets out, because yeah, I think it is a big issue. I think for Hawkman, it could certainly be an opportunity you could certainly use something like Hawkman to train a system to try and understand what parts of the image are likely to be reliable and what parts are not be. That would definitely have the possibility. I think in general, the types of issues I tend to see with systems that are using deep learning or artificial intelligence tend to be more related to either training. So the first big one I think I've seen is training on simulated data, because there are actually a surprising number of issues that can come from training on simulated data sets, even if you think it's very, very close to the real data set. And there are some really cool artifacts. Man, that sounds so sad, but I've been so excited by the artifacts I've managed to generate when we've used deep learning in using simulated data sets. There's some really interesting stuff out there. But it's only really starting to be understood because deep learning is such a new field still. And that's partly how I feel Hawkman can be quite useful. It's not a deep learning approach. It just really tries to go back to the basics and ask, is this stuff real? I think when we use deep learning, we're going to have to think about new ways in which we can verify data, and particularly what you do when you're using simulated data to do the training and then you use experimental data. And I think benchmarking is going to be a really big thing. And we're also going to have to think a lot about about how we verify for errors. And certainly tools like this that help you identify errors can be part of that. But I think discussing standards as a field and trying to understand what these algorithms are really doing is also going to be really, really important, because I think a lot of us don't fully understand what they're doing yet. And there are a lot of things which I see a lot now in papers, particularly, say, training networks on small numbers of images, which can be fine for certain types of thing you're training for, or certain applications, or when you are doing transfer learning. But in many cases, they will lead you to get artifacts. So we have this thing where we're using these very complex tools, and it's almost like the early days of deconvolution. We have to learn how to use them before we're going to be able to get accurate things out. But as you say, translational applications absolutely have to be accurate. So that's where we will have to be sure before we apply them.
Thank you, Susan. Maybe I get back to you with some data in how.
I would be really interested in that. That would be great.
Tell me what you think about it. Thank you.
I can see we also have a note on the decorrelation analysis that they're soon going to be publishing an update to do with image rendering that should help processing experimental data. So I'd be really excited by that. Yeah, it was a really fascinating paper, and I think it was very promising. But as I say, for us on the experimental data, we did find it challenging getting it working really well. So an update on that would be absolutely fantastic. Thorsten, did you have a question?
Hey, Susan, how are you? Fantastic tutorial. I really enjoyed that. Yeah, so what do you think you can reach in terms of high time resolution in live cells when using fluorescence proteins, especially given that you get more and more artifacts? Presumably, the higher the time resolution, the more artifacts you get, right?
Well, yeah, here's the interesting thing. So this is where it starts to become very important to understand the difference in terms of speed, in terms of types of structure, because if you go from a sample where you have, say, small clusters up to a sample where you have an extended 3D structure, that's a factor of 10,000 difference in terms of the amount of time it takes you to get a given resolution. So if you wanted to image small clusters fast, that's very, very viable. But if you want to image an extended 3D structure, it's pretty much impossible. So I think it's important, firstly, that the answer is going to be completely different depending on what type of structure that you're looking at. In terms of the actual imaging process, it comes down a lot to the performance of your label and how likely your sample is to keel over and die from being blasted with large amounts of light, which again is the issue that we tend to come up against is super resolution microscopy. So, we were able to do several minutes of high density imaging without any particular problem. But obviously at some point they do start to suffer from phototoxicity. We're moving now for our studies, we're moving out into the far red, which should obviously help. And in those cases, particularly, if you're able to operate near the cover slip, you don't have to illuminate a very large section. And, we find we can image for a few minutes at a time, which is about the amount that we can take in terms of data before it becomes completely overwhelming. Without any problem, the cells seem okay. But as I say, if you want to image a structure that has a lot of information across 3 dimensions, as compared to, say, the microtubules or the